So today we're going to talk about sleep in non-human animals, and we're going to cover quite a few different groups of animals, including simple organisms. We're going to talk about insects and fish and amphibians, reptiles, birds, land mammals, and marine animals, which are some of my favorite because they have some really interesting sleep patterns. But before we get started with exactly what we're going to do in terms of the different species of animals, first we have to make sure that we understand what is sleep and what is not sleep, right? And then we also have to define our criteria of what we consider to be sleep so that we know if it actually covers particular animals or not. So, first, beginning with some terminology, what are some terms that are actually not considered sleep? So, the first one is quiescence. And so, this is a period of inactivity or dormancy that is a quiet state that an animal might be in, or a species or an organism might be in, that doesn't necessarily look exactly like sleep, but it looks like the animal is in sort of a quiet state. Next, we have rest, and this is that reduced activity, but we don't see any loss of consciousness. Um, we don't see that the organism is as responsive. And so we see this is kind of a step up from quiescence, but still considered a restful state and not quite at the stage of sleep. Torpor is another type of situation that is not a state that is not sleep. And this is where we see less physical activity. We typically see decreased body temperature as well as decreased metabolic rate. And the reason why torpor is different from hibernation is because they are easily aroused from this state. We may even see this occur daily, we may see it occur seasonally, but typically the animal or organism is able to come out of this state pretty quickly, pretty easily. Whereas hibernation is where we see the organism not being easily aroused and in a state of inactivity, as well as metabolic depression. We typically see this when an animal is in a situation, an environment, for example, where there's not easy access to food, so they may have to hibernate for a period of weeks or months in order to conserve their energy. And so we see their metabolic processes slow down and allow them to be able to survive, for example, a winter. And of course, in this case, we do know that animals actually sleep during hibernation. So it's hibernation in of itself is not considered sleep, but we do actually see animals go through different stages and types of sleep while also in the hibernation state. So if we look at other species, and we want to see if we consider their experience as sleep, we really have to ask the question, is the state of sleeping the same experience as we see with humans, right? So we have to have a very specific definition of sleep that is going to include different animals. And it can't be the same exact experience as humans because not all organisms have the same kind of nervous system, for example. And so we have to ask the question, how do we look at different organisms and determine if they fall into the same experience or category as human sleep? We have to be able to distinguish sleep from other circadian changes in alertness. And typically, we see in mammals the suprachiasmatic nucleus in the hypothalamus in the brain typically is the one that controls circadian rhythm and body clocks. We have to be able to identify this and separate this out in other species. Just because we see an organism engaging in reduced activity, reduced alertness, we can't assume that they're in a sleep state. Here we've talked about other situations, quiescence, rest, torpor, for example, that we see reduced activity. And especially in hibernation, we see reduced activity and a change in metabolic rate. We have to be able to have a definition where we don't just assume that reduction in activity and alertness is sleep. So that means that what definition we do sort of fall on has to consider this, and it can't just be reduction in activity and alertness. It has to be more than that because we know that there's other states that are similar to sleep but are not physiologically the same. So let's now determine what kind of criteria do we have for different animals. So there are three major criteria that have to be met in order for us to determine that a particular organism does engage in sleep. Number one, we have to see a rapidly reversible state of immobility, as in, if the animal is or organism is in a seemingly restful state, in a rest state, okay, where we see reduced movement, they have to be able to get out of this very quickly. So like right off the bat, hibernation does not, meet the first criteria because in hibernation, an animal is in a state of immobility and cannot rapidly reverse out of that. Okay, obviously death is not rapidly reversible, and so that is different than sleep. The second criteria that has to be met is a greatly reduced sensory responsiveness. And this is an interesting one that seems to be a harder criteria to meet for some organisms because the responsiveness rate during different states are very different. And so we're going to talk about some examples where we would almost consider a particular organism having sleep, but the problem is this number two criteria, this reduced sensory responsiveness is questionable. And so we have to ensure that the animal, the organism, does in fact have a reduced responsiveness. Basically, their threshold is different when they're in this rest state. They're not as reactive, and it takes more to wake them up or more to get them to engage because their threshold is higher during this rest state, whereas it takes a lot more to get them aroused. And number three is that sleep must be homeostatically regulated. Okay, so if you have an organism that sleeps, if they don't sleep, you have to be able to see that the animal or organism wants to engage in sleep when they're deprived, okay? Homeostasis is essentially balance, right? So if an animal organism requires sleep and it doesn't get the sleep, we should see some deficits in some way. So anytime you lose sleep, okay, we have to see an increased drive for sleep to make that up, okay? So that's kind of step one in the homeostatic regulation criteria, okay? Any kind of deprivation or lost sleep, we have to see some increased drive to make that up, okay? So for like humans, if they only get 4 hours, but they need 7, you should see that that night that previously was deprived of sleep, that person is extra tired and really wants to go to sleep earlier because they need those extra hours, right? So then there's a balance there. So the second one is we need to see some kind of consequent sleep rebound, as in if an organism sleeps, let's say 8 hours per night, okay, and you deprive them of sleep for one night, their sleep rebound would mean that they would get more than their typical hours the subsequent night they're allowed to make that sleep up. So if you say an organism has to sleep 8 hours, that's their normal, okay? And then you deprive them and make them sleep 4, the next night we should see this number increase, maybe to, 9 to 10 hours. We should see a rebound. We should see the animal is trying to get more sleep and rebound that effect. And really the interesting thing is that if we humans, any animals, have no evolutionary need to sleep, if there's no purpose in sleeping, if we can survive without it, it's really one of the biggest mistakes in evolution, because if it doesn't serve a function, if it's not important, we're risking our lives in the case of humans sleeping 8 hours per night every night of our lives, as adults, and even more when we're younger. And so this evolutionary problem is that we really don't know why we sleep, but it must serve a purpose. It must serve a function for us to risk our lives every single night to do it. So this is kind of like a reverse way of thinking about it. Well, if we do this thing, it must serve a function. And we, of course, have a lot of guesses as to what function it serves, but we really don't know the absolute main reason why. And when it comes to looking at mammals sleeping, right? Sometimes we see, well, we're going to see a change in cortical activity, right? We're going to see a voltage change. But sometimes we see sleep being a high voltage change from wakefulness, or sometimes we see it as being a low voltage change from wakefulness. And this can get very confusing on which organism you're talking about, and can get kind of frustrating because you would want to say, well, if you have reduced brain activity, then that would probably fall in line with criteria number 2. But sometimes we see higher voltage activity because of what the brain is doing while that organism is asleep, and sometimes we do more things when we sleep than when we wake. And so this can be very confusing, and this can be frustrating because you would think if sleep in the brain all it does is rest, okay, then you would expect everything to have low voltage activity, but we know that that's absolutely not the case for animals. Now, when looking at when looking at sleep in other species, okay, we have very, very few studies of sleep, okay, in different animals. So we have over 60,000 species of vertebrate species, but sleep has only been studied in fewer than 50 of these species. And so we know very, very little about sleep in animals, and that's just because we just haven't done an exhaustive review, okay? We know that sleep deprivation in humans, so depriving a human of sleep, looks different than other species, okay? So for humans, we see that our temperatures tend to fall, we tend to gain weight. We see these different autonomic changes that in some cases are opposite in other animals. And so, we've only studied, roughly 50 species. We already know that there's really significant differences in how some animals sleep in terms of the amount of hours that they need and the physiological changes that we see during sleep. So it is such a difficult thing to nail down. It's such a difficult thing to study. So it's not easy to just say, oh, this animal sleeps or this animal doesn't sleep. Especially when our definition and our set of criteria have to be broad enough to cover all of these, different kinds of changes that we see across the board for different animals. Another really interesting thing is if we look at sleep in certain animals and use different techniques, we get different results. And that can be, again, a very frustrating thing because you're trying to study animals in a way that we have standards and we have the same sort of way that we study sleep deprivation and we study sleep in general. When we look at rats, for example, okay, so here we have an example situation. This is a particular technique called disc over water, okay, and essentially what happens is the rats are on this rotating disc. And so we have a control rat here that has not been sleep deprived. It's rested as normal. And then we have a sleep deprived rat over here. And the object is that the disc is moving, continuously moving, and the rats are continuously active. They have to be completely active because if not, they'll actually fall into the water. So for the control rats, fine. It's been, it hasn't been up all night or up all day. Rats are nocturnal. It's been, it's well rested. It's fine. And so it can walk on this platform and not be bothered by anything. Whereas the sleep deprived rat, the whole point is that as this rat is continuously moving, if this one pauses at all because it falls asleep, it'll actually get knocked into the water. And so it's a technique to demonstrate, to force a rat to stay awake when it's sleep deprived, okay? Because naturally the rat's going to just want to fall asleep. Now, after doing this for some time, okay, it doesn't take that long. Within 2 to 3 weeks of doing this for the sleep deprived rat, what we find is these same sort of things happen. First of all, we see an increase in body temperature. Now, remember, okay, body temperature falls for humans, and we see a weight gain. In rats, with this technique, only with this technique, we see an increase in body temperature, we see them eating more, and then we see their weight loss. So they don't gain weight, they lose weight. We also see a really interesting stereotyped pattern. So here you can see in this particular rat a very specific pattern we have nice shiny fur over here in a regular rat, and here we have kind of lackluster fur, it's not shiny, and also you see these like patches and lesions that occur in terms of fur loss and then lesions on the paws. And so we see this pattern of fur discoloration as well as these skin lesions. And eventually the body temperature is going to fall. And then within 2 to 3 weeks, this rat is going to die using this technique. Now, we cannot seem to replicate these kinds of results with other techniques. And so if we deprive using other ways, we don't get this same sort of set of results. Okay, so it's very interesting. Why is it that this particular technique with this particular animal, just with rats, seems to lead to these particular results? If we use the same technique in pigeons, it's not lethal. We don't see the same effects. We don't see the same metabolic and thermoregulatory changes that we see with the body temperature falling and weight and all of that. So what is it about this technique in these animals that seems to give us these really interesting and very lethal results? And we still don't have a great answer as to why it is that this disc over water technique seems to have such a detrimental effect on sleep-deprived rats. So how we study an animal is very important. Our criteria that we list is very important. And all of these can change the effects of and our conclusion if the animal is sleeping and how it's sleeping. Now, I'm going to pause here and I'm going to pick up on the next video. We're going to start our journey of sleep in different species and different organisms. So we're going to cover simple organisms, insects, fish, amphibians, reptiles, birds, land mammals, marine animals, mammals, and then we'll be through with this particular lecture. So come back around for part 2 so that we can dive into the different simple organisms and different species beyond this.